Hello, grade 12s. So now that we've gone through our basic introduction of the terminology related to genetics, it's time for us to go look back in the history relating to where genetics started. So if you think about it, if we're thinking from a historical perspective and we're thinking about people's names and who we need to remember, we are now moving on to the fifth person that we really need to know about. Because remember, we did Watson and Crick, and Wilkins and Franklin in DNA, so you need to know about them in terms of the discovery of the structure of DNA. And now the last one that's going to relate to DNA itself is Gregor Mendel, who is really part of the genetics side. So often when we look at Gregor Mendel, he is referred to as the father of modern genetics. And you're going to see why. So although it's, kind of like it's the inverted commas, he's the father of genetics, essentially, because his experiments that he conducted really laid the basis for our understanding of what happens in terms of genetics and understanding inheritance. How do we pass on these traits? And that's really where it was. So a little bit of background. So Mendel really was around in the 1800s. So he was kind of like born in 1820 odd. He was doing his research in the 1840s, 1850s, but originally he was a monk. So he was one of those rare combinations at the time where he was both a man of the cloth in terms of being a religious man, and he was also a scientist. So again, not like a scientist as we understand in these days, but he was wanting to understand, and he was more of a naturalist. So he wanted to understand how the world worked. He wanted to have an understanding of nature. So at the time, we didn't have, again, there was no concept of DNA. There was no concept of this structure of cells and all those kind of things. They didn't even have like a microscope really much in those kind of days. So really what they had this understanding of was there was these things that they referred to as factors that were passed on from generation to generation. So literally just from observing, the scientists around the time were able to understand by kind of looking at the animal kingdom, by looking at humans, that there was some way that certain characteristics or factors were passed on from generation to generation because, offspring looked very similar to their parents, maybe slightly different. Different families would have different factors that were related to them, but we didn't really have any sort of understanding of how this actually worked. So where Gregor Mendel came in was he did his very famous pea plant experiments. So literally, he was experimenting on pea plants, the peas that you eat kind of thing. And the reason why he chose pea plants was because they were very easy to handle. They grew fairly quickly for him to have an understanding. And there were very definite differences that he was able to look at to understand what were these factors that he wanted to look at. So these were the various factors that he was looking at. So he grew many, many, many different varieties, and he made them purebred. So for example, he looked at seed shape. Were they round or were they wrinkled? Seed color, yellow versus green. Looked at flower color. Were they purple flowers or were they white flowers? As well as a couple of other variations that he had here. So what he had to do in order to start this was he first had to make sure that they were all purebred to start with. So what do I mean by purebred? So purebred means that, say for example, I take the round pea shape. If I took a farm of pea plants that had round shapes, I would want to make sure that for several generations of when I bred different species, bred different versions, or not species, bred different plants together that had this round shape, I wanted to make sure that every single offspring that they had was only round-shaped peas, that none of them would ever have wrinkled peas. If I found that eventually I had only round-shaped peas, or wrinkled, or yellow, or green, or whatever, then I know that this is a purebred version of whatever this particular trait or factor is. And then he could start doing his experiments. So you had to do a lot of work first. He had to make sure that they were purebred first. Essentially, from our perspective, what was he trying to do? He was trying to make sure that they were all homozygous. essentially, as opposed to being heterozygous. You'll understand why. So yes, that is where this came from in terms of where he started. So let's take a little example to see what he did. So here's an example where he took a purebred purple flower and a purebred white flower. So all the rest of the plant was exactly the same. The pea shape, the pea color, the height, the shoot, the peas. The only difference between the two plants was that the one had purple plants and the one had purple flowers, and the one had white flowers. So that's really important because he was only wanting to look at the one difference that was occurring. So what he did was he allowed these to cross-fertilize. So this flower, this plant, he took the gamete from this plant and he let it fuse with the gamete of this plant. And what he found in the first filial generation, so filial generation, F1. So in generation 1, this F1, he found that all the plants were purple. So filial generation 1, they were all purple. And he found this for all the other combinations that he did. If he took non-wrinkled and wrinkled, he got it that one of the versions was only shown. If it was green and yellow peas, he would find that if I put the two different together, he would only get the one color, which was very interesting because this other color was then masked. It was then covered. It would never show up in the actual phenotype of this particular generation. So what he then did was he let these F1 generations self-fertilize. So he let these ones put the same, so the pollen and the ovary from the same flower, so not different flowers, he let the same flower. So he physically went and cut the anthers, he self-pollinated them to see what would happen. And what he found was that he would then have, on average, 3 purple flowers to 1 white flower. So it was a ratio. So on average, for every 3 purple flowers that he got 1 white flower. So this is this ratio that he found. Again, this was a prediction, or it wasn't necessarily, he could have had 10 in a row and they could have all been white. But then if he planted 100, ratio of 3 to 1. That's why it was very important to make sure that he did lots and lots of variations of this. He planted many, many plants, so his sample size had to be really big. So this was one of the main contributions that he gave us in terms of understanding the ratio of inheritance, this 3 to 1 inheritance. You'll see what that is in a little second. Okay, so understand, so he took these 2 purebred different ones, the only difference being 1, so this was flower color. He found that in the first generation, the first filial generation, only 1 of the colors was present. If he let that filial generation self-fertilize, he then found a ratio of 3 to 1 of the original color and to the opposite color. Right, so based on this, he was able to come up with his 3 main laws of inheritance. And we refer to these as Mendelian laws of genetics. So Mendelian, named after Mendel, and it's his laws of inheritance. So these are the basic laws of inheritance, what he came up with. And we're going to talk a little bit about them. So number 1 was the law of segregation. So this says that the 2 alleles for each gene are placed in different gametes. So essentially what he is saying is you will inherit 1 allele per parent. So when you form those gametes, each gamete will only get 1 of these alleles. And at the same time, you as an offspring will therefore only inherit 1 allele from each parent. So it's that segregation. We are separating the 2 alleles because I've got 2 different versions of it. Then, the law of independent assortment. The inheritance of one gene doesn't affect the inheritance of another gene. So basically what this is saying is your chance of inheriting any of these alleles or any of these variations is random. There is no, if I get this gene, I have to then get this gene. Independent means each and every single one of the genes is then independent of each other. We do know that this is slightly not quite the case now, because we know that several genes are located on one chromosome. So yes, if you get one gene and all the genes of chromosome 1, you will get whatever is all on those chromosome 1s. So although you have a random chance of inheriting them, if they're all located on the same gene, there is the potential that they are all semi-linked to each other. Whether you get the one from your parent or the one from your grandmother's side or from your grandfather's side, that is still independent. We also now know that there is crossing over that occurs. So that is very random. That random is there. So they are not necessarily, if I get this crossing over from one to another, that I have to have this gene go with the other one. No, there's lots of random chance. There is lots of independence. So essentially that is what this law is saying. And then the third one, which we've already kind of discussed with the terminology that we did, is his law of dominance. So when two different alleles are present, only one is dominant and will be expressed. So essentially this is showing the difference between dominant versus recessive. So if you have a dominant gene and you have a recessive gene, the dominant gene will always be expressed. So one will always mask the other one. And that goes back to his experiments where he had the purple and the white. He did his first generation. There was no white that was being shown. So that was the recessive. The dominant was then the purple. Please note that this is really only when we talk about complete dominance. So for your purposes, to make it nice and easy for you, we only deal with complete dominance cases, where if I have one version and another version, one will definitely be dominant over the other one. You do get cases where you can get something called co-dominance or incomplete dominance. Say, for example, you had a red flower and a white flower that breed together. You may then get pink flowers, which is a combination of the two colors. But we don't deal with those for you at your matric. We only deal with complete dominance. If you want to go and learn a bit more about incomplete dominance, go do some reading. It's very fascinating to understand how these different things relate to each other, and that really is important for us to understand. But for you guys, we are only focusing on complete dominance. So Mendel's law really here does, if there's two different ones, one will be dominant, one will be recessive, and then that's it. Okay. So let's just have a little quick summary of what we've done in terms of Mendel. So we know that he is kind of considered the father of genetics. We know that he did, so how did he get his knowledge? He did plant experiments. That was physically what he did. What did he do? He took purebred versions of things such as the pea color, the pea shape, the height of where the flower is, the flower color, all those kind of versions, and he then created hybrids. And then after the first generation, he made them self-pollinate. So that was essentially what he did. What did he provide for us? Well, we then understand the ratio of inheritance. And the ratio particularly that we are referring to here is when there are heterozygous combinations. So when heterozygous combines with heterozygous, we are going to have a 3-to-1 ratio of whatever is the dominant to the recessive. So that was the ratio of inheritance. And then he also provided us his 3 laws of inheritance, which being the law of dominance, the law of independent assortment, and the law of segregation. So you need to know what all those 3 laws are. So just as a side note, you need to be very careful when they ask questions related to any historical related ones. You have to be very careful about how the question is constructed. Because if the question, there's a difference between asking a question of who is Mendel, what did Mendel do in his experiments, what are his 3 laws, what information did he provide for us for his understanding of inheritance, which would be this, versus other things. Please make sure you read the question very carefully. One last thing that I'm just going to deal with here as a sort of AIM 3 perspective is the AIM 3 section that talks about how knowledge, let me get spelling, is accepted or reject it based on preconceived ideas. So what this is referring to is, so remember, Mendel was really around in the 1800s. But what happened was that his work was not really accepted by the scientific community. What happened was that it was only really in the early 1900s that his concepts were actually re-picked up by scientists to say, hey, he actually has some really good ideas. So although Mendel had died by this stage, his ideas then lived on towards a time where it was much more generally accepted. It kind of harps back a little bit to, like, Galileo, we're not a flat earth, we don't revolve around the sun, we revolve around the sun, that nothing revolves around us. It's kind of those ideas. So really just have a conversation with yourself to understand. So imagine if someone comes to you with an idea now. So, whatever it is, if we have a preconceived set idea, we now know that we try and base it a lot on scientific communication that we are doing at the moment. So what we are then needing to consider is, how do we change our ideas based on new knowledge that is coming in? Do we reject something immediately because it doesn't work with our status quo? Are we open to these potential ideas because it may challenge something that we already know? Really, that's a conversation that we can have to yourself. That's quite a really intense AIM 3 style question that you can have. Okay, I'm going to leave it there. So that's really kind of like the history relating to genetics and everything related to Mendel. Please make sure you go over all the different things. Think about the different questions that can be asked to you. Make sure you can word your things correctly, because your wording becomes very important here. And yes, we'll then carry on. Hope you're enjoying the studying, guys. Keep up the good work. Thanks very much.